Funding for this podcast is provided in part by the IPM Network through the USDA National Institute of Food and Agriculture and by various commodity groups through checkoff dollars, including the United Soybean Board, National Corn Growers Association, and Cotton Incorporated.
The I See Dead Plants podcast shares the stories of people and plants, pests and pathogens, and the conflicts among them. Join us as we speak to the folks who attempt to solve complex problems with plants through pest management research. I See Dead Plants is an extension-based podcast created by the Crop Protection Network and the North Central Integrated Pest Management Center and hosted by Ed Zaworski.
Welcome back to the I See Dead Plants podcast. As always, I'm your host, Ed Zaworski. Today, we got a fun one. It's no secret that I'm a nematode lover, and I've got Dr. David Shapiro-Ilan with me here today from the USDA, and we're going to be talking about a special kind of nematode, one that is a parasite of insects, an entomopathogenic nematode. And as always, before we get started, I'm just going to do a little bit of background info on David for you folks listening. He got his bachelor's in biology at the University of Michigan in Ann Arbor, followed by a master's in entomology at Louisiana State, followed by a PhD in entomology at Iowa State here, and he's currently a research entomologist with the USDA-ARS. All sound about right, David?
Yep, that's it.
So, David, entomopathogenic nematodes, what are they?
So, as we know, nematodes are roundworms, so not to be confused with other worms like the segmented worms, the earthworms, or flatworms. They have their own phylum. Entomopathogenic nematodes are those that kill insects, and there are other nematodes that kill insects, but what's special about entomopathogenic nematodes is they kill the insects through a really fascinating partnership with a symbiotic bacteria. So there's two families of entomopathogenic nematodes that everyone recognizes, the Nematode and the Heterobdididae. And the nematodes are associated with Xenorhabdis bacteria, and Heterobdidid nematodes are associated with Photorhabdis bacteria. And it's called Photorhabdis because it makes the dead insect glow in the dark. The Photorhabdis bacteria are luminescent. So those are what most people consider entomopathogenic nematodes. There are some other species in the genus Osseus, which some will also consider to be entomopathogenic because they do carry bacteria, but the relationship with Osseus species and their bacteria is not nearly as specific as the Steinonema and Heterobdidus, the traditional entomopathogenic nematodes.
Where can these nematodes be found? Are they in the soil, in plant tissue? How do they become associated with insects?
They occur naturally in soil and actually occur in every continent except Antarctica. And they're found in natural areas like forests and so on, but also in agricultural systems.
So you mentioned there is this relationship between the nematode and the bacteria and the insect. Can you kind of walk us through the process of how that all works and how they actually end up killing the insect?
Right. So the nematodes only exist in the soil, in the natural environment, as what we call infective juveniles, or IJs. That's the only free-living stage. And when those infective juveniles come upon an insect, they'll attack it. Some of them are kind of ambush foragers. They'll sit on the soil surface and wait for an insect to come by and then ambush it. They can even jump, like some nematodes can jump an amazing distance for a tiny nematode. So those are the ambushers. Others are more like a cruiser strategy. They'll move through the soil like in a search and destroy mission and try and find insects to kill. Some forage in kind of in between pattern between ambusher and cruiser. Once they find an insect, they're going to enter that insect usually through natural openings, mouth, anus, or spiracles, but they can also sometimes go through the cuticle of the insect. Once inside, they're going to release their partner, the symbiotic bacteria, And those bacteria carry the bulk of the virulence factors, but the nematodes also contribute to killing that insect host. They have their own toxins and immune suppressive factors. So between the two, the bacteria and the nematode kill the insect usually within 24 to 48 hours post-infection. And then both nematodes and the bacteria will proliferate in that host, and you'll get 1 to 3 generations of nematodes reproducing in that dead insect, and then you really get like an explosion of new nematodes, new infective juveniles coming out of that insect and ready to go find another one to infect. So you could get tens of thousands or hundreds of thousands of new nematode progeny coming out of a single insect host.
I'm just kind of curious along those lines, David. Is this a necessary relationship between the nematode and the insect? Can the nematode still reproduce in absence of the insect, or with these particular species, do they need each other?
So
well,
the
not each
nematodes,
other.
yeah, the insects don't want the nematodes,
Yeah.
but the nematodes in nature are obligate parasites. They will only reproduce when they find an insect. So when they get in the insect, they molt from that infective juvenile to adult, and then they mate and reproduce. The relationship between the nematodes and the bacteria is a true mutualism, so that the bacteria, they can't survive in the environment, except one rare species. They can't survive in the environment without the nematode, so they depend on the nematode to carry them from one host to another.
I'm kind of curious too. I mean, well, there's two things I'm curious about. I'll start with the first one. Are there specific insects that are more susceptible to entomopathogenic nematodes?
It's kind of hard to generalize. There are a lot of beetles, say, that are the Coleoptera that are susceptible to nematodes, various weevils, various scarab beetles. But then you'll find some beetles that are not susceptible, like the Prunus root borer is really, we've tried to put so many nematodes on Prunus larvae, and it seems they're like a tank. Nothing can kill them. So it's kind of hard to generalize. Yeah, but these nematodes are used commercially, and they're used against a wide variety of insect pests crossing many different insect orders.
Got it. Yeah, I was just wondering more or less like, if the insect itself isn't coming into contact with the soil or other insects that are infected, like would a housefly ever be attacked by entomopathogenic nematodes, for example?
As a larva, maybe when it's like in the soil or in a semi-manure environment, but when it's flying around, not too likely. They are soil-dwelling organisms, the nematodes, though we do apply them sometimes above ground, but we have to have special formulations to help protect them from desiccation and UV radiation, so that they can kill, for example, insects that bore into the tree. Otherwise, if we don't use those protective formulations, most likely the nematodes will die before they can ever reach the insect pest.
You mentioned some of the coleopterans being prime targets for nematodes, let's say. Can the nematodes affect adults as well as larvae, or is it mostly the larvae that they're targeting?
They can affect both, and it's really going to vary depending on the host species, which is more susceptible. For example, we found in pecan weevil, the adult weevil is much more susceptible than the larva, though in a number of other insects, it's the larva that's going to be more susceptible. But again, you have to have a mechanism for the nematodes to reach that host. So pecan weevil adults also spend some time in the soil, so they're very easy targets.
So it's more about the nematode getting to the target, more or less, then.
I think so. And then, the insect will also mount an immune response, and maybe sometimes the larval stage is more adept at fighting off the nematodes than the adult. So there's a lot of factors involved.
Interesting. So, David, I was recently in a seminar of a graduate student here at Iowa State that he was working with entomopathogenic nematodes, at least for part of his master's project. A question formed in my mind while I was sitting in this seminar, and I figured this would be the opportune time to do it. If you're trying to target, a particular insect in a system with entomopathogenic nematodes, I was kind of curious, like, do you ever have kind of off-target, unintended effects of these entomopathogens on insects, beneficial insects, let's say? And I was just kind of curious, like, how this was a question I asked earlier, more or less, about what insects are susceptible, but is there any kind of host specificity between a particular species of nematode and its host, or are they just kind of whatever they can get their hands on?
So most of the entomopathogenic nematode species have a broad host range. For example, Staudinemocarpocapsae, which is probably the most studied and maybe about the most used nematode commercially. In the lab, it can kill like at least 200 different insect hosts. But in the field, it's going to be more limited. Most of the nematodes, though, can infect insects across several orders. There are just a couple of exceptions, like Staudinemopathogenic really focuses on the Orthoptera, like especially mole crickets, whereas Staudinemoscarabi is kind of specialized to scarab beetles. But as far as impacting beneficial insects, generally the thinking is that it's either going to be negligible or minimal. Part of that is the susceptibility. Part of it is, are they really going to reach those insects? So we've studied lady beetles and their susceptibility. You could take lady beetles into the lab, and if you pile on enough nematodes, it's going to kill the insect. But to what extent is that going to impact the insects in the field? Pretty much the field studies that have been done, and there have been several, show that the impact on beneficials is minimal or negligible, and if there is an impact, that the beneficials will be bouncing back pretty fast. So we really do see a significant difference between an application of beneficial nematodes compared to, say, a broad spectrum chemical that is likely to take out a lot of those beneficials.
This is actually the perfect bridge, because I definitely wanted to talk to you about how are these things, what are the applications, how are they used? So I'm envisioning in my head, somehow you're broadcast spraying them onto a target crop. But yeah, just in general, how are entomopathogenic nematodes kind of used in a field situation?
Yeah, so nowadays a lot of the formulations are very amenable to use. So you may get just a packet that you throw into a spray tank or into an irrigation system. And most standard agricultural equipment can be used for applying the nematodes. You probably have to take out some of the screens, especially fine screens in the sprayers, because they might shred the nematodes. But otherwise, they can be sprayed or put through irrigation systems, just like other pesticides. And they can handle quite a bit of pressure. That's generally not an issue in some of the high pressure sprayers as well. They've been applied through aerial application as well, even.
So if somebody's going to apply some entomopathogenic nematodes on their field, are they buying like a solution of nematodes, or is it like a gelatinous mixture? What does that kind of look like?
There's some various formulations these days. A lot of the bigger nematode producers are either going to put them in sort of a vermiculite formulation, so it's just like a packet that you just granular that you pour into the spray tank, or maybe based on diatomaceous earth. There's some that will put it into kind of a gel that you have to wash the nematodes out and take that extra step, or in a sponge and wash them out. But generally, they're very much user-friendly formulations.
Yeah, that makes sense. If it's difficult, adoption would be more difficult. Uh, how are these things? How are entomopathogenic nematodes produced, David? Like, are they cultured more or less on the insects that you would be targeting? How are they produced?
So, really, in several fashions for commercial production, they all produce both in vivo and in vitro. So, in vivo would be infecting large numbers of insects and then basically harvesting the nematodes from those insects after they've been infected and the life cycle of the nematodes complete. Um, then you also have in vitro, either solid culture or liquid culture in sterile fermentation. So you can have large, even up to 80,000 liter fermentation tanks where the nematodes are being grown, or solid culture where they're grown in like sterile sponge. Most of the commercial nematodes now are grown in liquid culture, but
Hmm.
there are still commercial entities that produce them in vivo. And we've done a lot of work improving both of those systems for in vivo, really making it more streamlined so there's not so much labor involved, so that it's bringing the cost down and bringing the cost down for the farmers and the producers. And then in vitro work we've done is to improve the media to get better quality nematodes. Recently, we developed a system where the farmers can produce their own nematodes, and we in the process of completing a Jove article, Journal of Visualized Experiments, showing how that process works, and it's been published. So the farmer kind of just follows the instructions, and they could produce their nematodes continually for their own fields if they want to.
Oh, interesting. Interesting.
Yeah.
Along those lines, are entomopathogenic nematodes costly to produce, or are they costly for someone to purchase? I mean, obviously we don't have to talk in exact numbers, but are they, yeah, are they costly?
Usually compared to, you know, broad spectrum chemical insecticides, they're more expensive.
Mhm.
Sometimes considerably so. But it depends a lot on the particular target and the use. If you can target the spray of the nematodes, then you're using less and they become less expensive. For example, against peach tree borer, you only have to apply right around the tree because the insect's straight down in the taproot. And that really reduces how many nematodes you need per acre. So you may have really a comparable cost compared to some other approaches, chemical insecticides, and less expensive than, say, mating disruption.
Mm.
Also in citrus, to control the diaprepi citrus weevil, you're only applying under the canopy, so the costs much more amenable to the growers. And I would say nowadays, when you're having more target-specific chemicals, or they become more expensive too. So I think the gap is decreasing. And there's other ways we explore how to reduce the cost. And one of those that's been looked at recently is, and that we're exploring as well, is the nematodes being more persistent. Can they go from one year to the next without having to reapply? And we're starting to see that there is potential for that. And some other people are studying that as well. Myself, with Kyle Slusher in Texas A&M looking at pecan weevil, saw that we did see carryover, and even at lower rates. So this is something that really has to be explored further. Can the growers get more bang for the buck and apply one time and get carryover to the next year? Because the previous dogma was really that, oh no, the nematodes, they're not going to last from one year to the next. You'll find them in the field, but not at the levels you need to control the pest. But that's being
Mm.
challenged now, and so that's being researched further.
Yeah, you beat me to my next question, and that's I'm happy to hear that answer because I was thinking, yeah, along the lines of having the cost being a little bit elevated. Yeah, if you could recoup a little bit by having them persist, at least at some level, That makes the whole equation a little bit more interesting as far as kind of return on investment situations.
I was going to say there are other mechanisms that we're looking at to reduce the cost, such as using kind of boosters like nematode pheromones that enhance their behavior. They kind of turn them on, so you may not have to use as many nematodes, or they're more efficacious. The nematodes kind of talk to each other with these pheromones, so we're basically directing their behavior by telling them, now it's time to go find an insect and kill it. So working with a cooperative industry partner, Ferrinum, we use these pheromones and enhance the behavior and hoping to reduce costs for the grower in that manner.
Interesting. David, I mean, you started to talk about some of the situations in which these nematodes are used. Do you want to give us some situations where nematodes, entomopathogenic nematodes, have been used effectively?
Right. So definitely a big success story has been Florida citrus against the citrus root weevil, and nematodes were applied practically like the solution, not much else going on to control those pests for a long time. I'd say now the farmers there are especially worried about the greening disease, so nematode use may have fallen off some, but that was a success story. And then you also have greenhouse crops, very big target with fungus gnats and thrips, and also fungus gnats and thrips in other industries. Mushrooms, the fungus gnats being a primary pest and an excellent outlet for the nematodes. And then also to some extent they have use in orchard crops, as well as even sometimes some corn.
At this point, David, I kind of want to move into wrap-up questions that I like to ask at the end, kind of these overarching philosophical questions. But before I get into that, I always kind of like to pause and ask you, I write these questions ahead of time. I don't always know if I'm asking all the questions that I need to ask to inform my listeners. Is there anything that we missed with regards to entomopathogenic nematodes that you'd like for people to know about?
I mean, in general, they're just fascinating organisms. The whole thing with the mutualism that they have, and symbiotic bacteria and luminescent bacteria. So they're really very interesting to study, and they're really, I think a potent biocontrol agent. They're used against a lot of economically important pests.
That's a good, wonderful summary. So let's get into these wrap-up questions. I always have kind of fun with them. Hopefully you do too. So the first thing I have for you is, what do you think that entomopathogenic nematodes will kind of increase in their use over time, expand maybe in their applications? What do you think?
I do. As we develop new technology, better technology with the nematodes, I can definitely see their use expanding, as we find better nematodes, or we even breed better nematodes to use in agriculture. That's going to enhance their use, and there's pheromone use and other boosters that we talk about to increase efficacy, better formulations. All of those things I think will make nematode use more attractive to the growers.
Along those lines, what are some of the hurdles that people who want to use entomopathogenic nematodes, what are some of the hurdles that they run into that perhaps are being worked on?
Right, and we talked about cost and how we want to, improve the cost, but I think also I think one barrier is knowledge about the nematodes. Like any biocontrol agent, that they are relatively easy to use. They're live organisms, so you can't, put them in your spray tank and then go off and catch a meeting for half of the day and come back and they're cooked in the spray tank or something. They have requirements in terms of temperature, optimum, and also, aeration. You can't leave them in the tank so long that without aeration. When you apply them, they have to stay moist for a certain period. Applying irrigation or keeping the soil moist is important. one kind of broad barrier to wider use is the nematode sensitivity to UV radiation and desiccation, so that if you apply above ground, this can be a problem. Like I mentioned before, if you apply nematodes to a wood borer, say, they may die before the nematodes die before they get in and kill the host. So we have been developing protective formulations like a fire gel that's used to protect forests or structures against fire. Also stays wet a long time. So we found it keeps the nematodes moist for a long time so they could get in and kill the host. And we're developing other formulations like even nanoparticles so that each little nematode gets a coating, like it has its own little suit of armor that protects it from UV radiation and desiccation. So these kind of technical advances as we move along, I think, will overcome the barriers to use.
Excellent. David, if you were speaking to a farmer who is kind of considering trying some of these EPNs, what would your message to them be?
I would say that they need to consider, of course, for themselves what they see as the cost benefits. I would encourage them, based on the literature and what I know, to, consider the efficacy, and, if it's a good fit, then I would be encouraging.
My last one for you, David, is kind of a fun one that I've been doing since the inception of the podcast. I like to ask people, if you were talking to a member of the public, and I like to frame it up as my mother, she lives in a Chicago suburb right off of Lake Michigan. She has no clue that entomopathogenic nematodes probably even exist. How would you explain this to someone like my mom?
I guess I would say that you have these little tiny worms that they look, if you have a glass of water, they'll look like little dots. If they're in the soil, you won't see them at all. But they're really potent insect killers, and they have a really interesting biology in that they're closely associated with this partner, which is a bacteria, and together the bacteria and nematode kill the insect pest, and these little worms are available commercially, and we're working on ways to improve their use and make it more profitable for the farmers.
Perfect. I think that's a message that a lot of people could get behind. So with that, David, I like to leave here time at the end for you to make any acknowledgments you want to, and you to point people to any resources you might want to point them to. You have the floor.
Yeah, I would definitely want to thank USDA-ARS and my lab for all their contributions, all my collaborators. We have collaborators in many universities, so I can't really name them all, but me and Ed Lewis recently published a book on entomopathogenic nematodes. So Shapiro-Ilan and Lewis, if you want to look up that title, Entomopathogenic Nematodes as Biocontrol Agents, then you'll see everybody practically who's working on it. So we just passed our 50th anniversary of studying entomopathogenic nematodes, the discipline, and we're excited to see it go forward.
Well, I can't speak for everybody who's listening, but I know I'll be looking up that book. So with that, folks, I hope this was informative for everybody listening. I hope you learned a lot about entomopathogenic nematodes, or EPNs. And thanks for listening, and we'll see you next time. Dr. David Shapiro-Ilan is thankful for all those he collaborates with on research investigating the use of entomopathogenic nematodes as biocontrol agents. There are too many of them to list them all, however. Among his current collaborators, he would like to acknowledge Dr. Ed Lewis at the University of Idaho, Dr. Fatma Kaplan at Farnham Incorporated, Dr. Michael Taves at the University of Georgia, Dr. Apurba Barman, University of Georgia, Dr. George Mbata, Fort Valley State University, Dr. Angel Helms, Texas A&M University, Dr. Tracy Lesky, USDA-ARS, Dr. Jaime Pinheiro, University of Massachusetts, his recent former postdocs, Dr. Kyle Slusher, Texas A&M University, Dr. Germaine Perrier, Mississippi State University, Dr. Xiaohui Wu, the Ohio State University, and Dr. Colin Wong, MGK Incorporated. Also, his current lab members, Stacy Bird, Kate Anderson, Destiny Absher, Dr. Abigail Kropp, and Dr. Sarish Gulzar.
I See Dead Plants bridges the gap between plant science research and the impact it has on our daily lives. It is brought to you by the Crop Protection Network and the North Central Integrated Pest Management Center, which are both partially supported by the USDA National Institute of Food and Agriculture. For more information on today's topic or other pest management issues, check out our show notes, cropprotectionnetwork.org or ntipmc.org. Remember, this information is for entertainment purposes only. The statements made in this podcast should be interpreted within the limited context of the particular topics being discussed. Contact your state extension program for local information on pest management. For the full non-discrimination statement or for accommodation inquiries, go to cropprotectionnetwork.org/about. Music written and recorded by fellow plant pathologist Sean Toporek. Thank you for listening to I See Dead Plants.